Hello, everyone, and welcome to the latest episode of This Week in Science. We are here again to talk about all the stuff that's been going on in science for the last week. We know there's a lot more out there. But we're going to stay in science land. So, as you know, if you are a regular visitor, viewer, enjoyer of This Week in Science, this part right now is the part where I go, hey, we're live, ta-da! And when we make mistakes, that's going to get cut out, and the podcast hopefully will not contain those mistakes, thanks to our wonderful editor, Rachel. And if you want to subscribe right now, there's like YouTube, Twitch, Facebook, those wonderful places. We stream live Wednesdays, 8 p.m.-ish Pacific time. There should always be a little slush time there on the end. And yeah, hopefully the show goes great, and I hope you enjoy it. Justin, how's your microphone? Tap, tap.
Oh, good question. Yeah, let me just...
It's a little echoey.
Yeah, it's probably on the wrong thingy. Yeah, you're so good.
But I forgot to... I knew it was there, but it was in the back of my head the whole time as I was getting ready.
What would I do without you? There we go. Now it should be very voicey.
It's way better. Thank you very much. Yeah. And my cat just jumped down in a little small space between things that the internet is involved. There's cords back there. So
if the show suddenly disappears, it's the cat's fault.
It's the cat. It's not me.
It'd be the cat.
I had nothing to do with it. All right, everyone. Welcome. And thank you for joining us for this episode of TWIS. What are we doing now? Did I make a new show? TWIS. This Week in Extra Sausage. Right here. Okay. Beginning of the show. You ready? Yeah, we're going to start the show in 3, 2. This is TWIS. This Week in Science, episode number 993, recorded Wednesday, November 13th, 2024. Science is better when done together. Hey, everyone, I am Dr. Kiki, and tonight we will fill your heads with questions, challenges, and restoration. But first...
Disclaimer. There is something disruptive taking place in science, something that is already altering the pace of research. New predictive models for protein structures, turning months of research into just hours. Whole genome analyzers that can identify single nucleotide variants in just minutes. Gene therapies that rewrite faulty human code, stem cell treatments that reverse the effects of aging, diseases once untreatable becoming manageable, and the manageable ones becoming cured. Our understanding of everything is being disrupted by more and more knowledge. Better knowledge, better tools, better medicine, better life. But there is one thing that is unfortunately not getting better. One thing upon which we cannot improve. One unchanging fact that no matter how we might try, continues to remain the same. Despite all of our knowledge, we still want more. This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki.
And a good science to you too, Justin, and everyone out there. Welcome to another wonderful episode of This Week in Science. We're so glad that you're here with us again for another week. My cat has joined us from underneath the couch, internet cable space. That's exciting. It's so amazing. There's a lot going on in this world. And we have a lot of science for the show this week. I have stories about solving problems, testing things, and being human. Justin, what did you bring?
New paper on old theory of the universe that might be coming true. Yeah.
Everything old is new again.
Yeah. Restoring eyesight with regenerative medicine. A huge study with a very small sample size. We have, is the UN a scam?
No, but we'll talk
Well,
about that.
wait till we get to the story.
And what does it have to do with science?
Everything, because
I know.
it's the COP 29, 30, whatever they were on now.
Right. There's a big meeting happening.
And then finally, there is a whole field of science I didn't even really realize existed. Diagnosing diseases from ancient artwork with a little bit of maybe something extra. Yeah.
Now I am very interested in, anything extra is extra. Like the first part was exciting, diagnosing diseases from artwork. That's really cool. Okay. I have so many questions. Can't wait for the stories. I hope everybody is feeling the very same way that I am. This is exciting. Something to look forward to. There's good in the world. There's so much curiosity out there, and we can carry forward with all of this. And as we jump into the show, I do need to remind you that if you are not yet subscribed, you can find us streaming live weekly on YouTube, Facebook, and Twitch, 8 p.m. Pacific time-ish. And you can look for This Week in Science, TWIS Science, on places like Blue Sky, Mastodon, and Facebook, others. If you can't remember all this stuff, TWIS.org is our website, and you can find show notes, links to stories, other exciting things there. So exciting. So exciting. But we love your subscriptions, and we'd love you to share the show right now as we start listening. All right, let us dive in. I know you're going to be talking about some of these climate change, UN, COP issues later, but I want to start out the show with a bit of the solution side of climate change and, where things are going. So big question historically is, okay, now we know there's a link, CO2 and temperature. The higher that carbon dioxide concentrations get in the atmosphere, in the oceans, the more the oceans acidify, the more the atmosphere heats up, and our temperatures increase globally. This is average. This is not just everywhere locally.
Kiki taking us all the way back to the 1800s when this was first understood.
Yeah, it was first understood a long, but I have to set a basic understanding, right? This is where we're coming from.
Yeah.
But this CO2, we keep emitting it. The Industrial Revolution onward, we've been like, smokestacks. Look at me making things. All my cows, everything I do. Let's put more carbon dioxide in the atmosphere. So how are we going to deal with that? What are we doing? There are real practical engineering questions that are involved right now. And there's a new study out through Massachusetts Institute of Technology, and it is published in Nature Communications right now. And this particular study, these engineers, graduate students, included in the team, Michael Nietzsche and Sanjay Garamella, Jack Lake, who was a graduate student in 23. They've been trying to look at ways that actually scale and are economically viable. Because one of the things is like, if money and energy and scaling of technology were no issue, we probably would have solved some of the problems that we're up against already. However, we have a big planet.
Seemingly. But this has also been one of those things like, well, we could plant trees.
No, you can't just plant trees. No, there's...
Hang on. I'm not saying that.
You're taking me away from my stories.
No, I'm not. I'm reinforcing the story. Like, one example was like, so we have all these now tree planting commitments by countries. The problem is, to plant enough trees to offset, you would have to use up all of the farmland.
It depends where they are.
Literally cover
what kind of trees they are. There's so many factors, right?
Yeah, like that is not in a standalone solution. So that's what I mean about the scalability. It's like, yes, if we had a bigger planet, that could work.
Right.
That's all
And
I'm
if
saying.
and if all the billionaires, trillionaires on the planet decided to put their money into trying to
You need like
harvest,
6 of them.
yeah, who made 60
6
billion more
people.
dollars actually after the
6
election? But
people.
um, ethylene, it's a product that is a carbon dioxide or carbon-based product. You can convert carbon dioxide fairly easily into ethylene, but it takes chemistry, it takes electricity to make it happen. Ethylene can be used to make fuels, plastics, all sorts of stuff that petroleum used to do today. So if we wanted to get away from fossil fuels and actually start reusing our emissions, we've got so many emissions. We could take stuff out of the atmosphere. We would be fine for a really, really long time if we could meet or beat the price that ethylene sells at currently, which is $1,000 per ton. Additionally, if to be able to do that, we can create an efficient process that the catalyst and the solution in which it occurs is efficient enough, it doesn't degrade too quickly, so that you're able to get a high enough concentration of the ethylene to make it over a big enough scale to make it worthwhile. So the researchers at MIT who are publishing this particular paper that I said is in Nature Communications, they have done a number of tests in which they first wanted to make sure that when you're doing carbon dioxide emission kind of harvesting, you have to take the carbon dioxide and grab onto it, and it's part of an electrolyte solution. And the electrolyte solution then has to go through an electrode surface, and there's usually water involved. But you don't want the water to interfere with the reactions taking place, because then it just disrupts a lot of stuff. Water itself is universal solvent. It can change the way that the chemistry takes place. So you want a really hydrophobic surface. They basically use Teflon. We don't like Teflon that much for our frying pans anymore, but maybe Teflon being used in this particular filtration and emissions harvesting scenario, it may work. I'm not saying this is good, this is bad, this is what they've done. PTFE is the compound that they've used. Very hydrophobic. They are able to put it together. There's a lack of conductivity though, so they need the conductivity to actually get the electrodes and the catalysts to work to like move the carbon dioxide through to create the ethylene. So they wove copper wires through sheets of PTFE in fine enough mesh, in fine enough grain that it was hydrophobic and also conductive at the same time. They showed that it could work at little teeny tiny scales of electrodes, which can be teeny tiny, but on an industrial scale, we're talking big electrodes. This really wouldn't be that efficient. And they saw when they tried to go bigger and bigger, there were a lot of other breakdown issues and metabolites and other things that were parts of the chemical solutions that you wouldn't necessarily want in the end. So the way that they have created their situation, it's like creating lots of little teeny tiny spaces, modular little spots within a fabric that little electrodes can connect to, that are extremely conductive, efficient, able to work, and would probably scale up very well. They did not scale it up to industrial specifications for sure. But the lab versions, they went from like tiny little versions to 5 centimeters bigger, which actually, when you're looking at a chemistry lab, engineering lab, this is pretty big. And they say it is the first PTFE based electrode, which has gone beyond the lab scale on the order of 5 centimeters or smaller. And it is the first work that's progressed into a much larger scale and has done so without sacrificing efficiency. This weave with copper and PTFE can be integrated into existing manufacturing processes. There could be large rolls made. Like, this is something that could be implemented. There's potential there. So the exciting thing is the ideas are there. People are working. They are looking, and as these graduate students, then the researchers who run the labs say, they're starting with a mindset of thinking about how the solutions are going to scale, as opposed to simply the science. behind making it work. So it's not just the science, it's also scalability. And that is that mindset is where they're starting now. And I think that's great. And beyond that, also the National Oceanographic and Atmospheric Association, the White House, and others who have been working together, the Biden-Harris administration, they've all released the National Marine Carbon Dioxide Removal Research Strategy this last week. This is to advance safe and effective research on the benefits, risks, and trade-offs of marine carbon dioxide removal. And this, to me, is an interesting and potentially problematic, but also exciting direction for all of our research to go. And as the current administration and this report have set things out, they're looking at biological and non-biological solutions. So can we create pumps that create artificial upwelling where they take bacterial-rich, nutrient-dense water and bring it up to the surface faster than would normally happen with usual day-night cycles? Artificial downwelling. Can you take the water at the surface, push it down to the colder water down below to be able to transfer the carbon dioxide more liberally and evenly throughout the oceans? Direct ocean capture, I think, is exciting because that is like, let's take the carbon dioxide out of the oceans as opposed to continuing to rely on the oceans as just a place for all the carbon dioxide from the atmosphere to get soaked up, because that sponge is going to get full. Yeah, there's the other, which is definitely biological microalgal cultivation. So getting lots more algae, photosynthetic organisms in the water doing the work to basically capture and store. Basically, they capture the carbon dioxide and then they die and they go to the bottom of the ocean and become the sediment on the bottom of the ocean. That's where the carbon dioxide is left. But then non-biological processes are ocean fertilization, which can be, putting more iron, more nitrates, more different products into the ocean to enhance the ecology of different areas. And so the question is, will ocean fertilization lead, is it going to mess things up the way that our fertilization and agriculture has led to algal blooms in some of our gulfs, our bays, our lakes, different areas? And then we have ocean alkalinity enhancement, where they're actually talking about changing the pH of the ocean so that there is more room, so the sponge can be maintained.
All I want to say is they need to stop it.
Right.
Like none of these ideas are the solution. Like, I'm sorry, no.
It's part of it. None of them are the solution. I mean, the reality is there is no one solution. Unless we just figure out, how are we going to use less? How are we going to emit less carbon dioxide? How are we going to? The engineering approach to harvest our emissions before they become emissions, to turn them into useful products. Those are really useful things that might, they'll negate or reduce the current growth that we have and the trajectory that we're on. But nothing is going to be the one. There is no silver bullet. It is everything and all the things. But I really am worried about the kind of terraforming our own planet stuff. I mean, we've done enough of that. But,
I mean, like, first of all is the scalability thing that we were just talking about.
Mm-hmm.
The ocean's really, really big. I don't feel like we're going to have a positive impact on that experiment in any of these models that I'm looking at right here. I would be curious to see, though, like, because
these are not all of the solutions. These are some of them, and these are just places to start looking, and they are not going to and the way that the report has been put out, it's not like, yes, we're going to do it.
No, I hear you. I'm even wondering, though, if there's a good enough reason to pour resources in this direction. Altering the chemistry of the ocean is
How about let's not do that.
Yeah. I feel like you're like not following the thread of how we got into this problem in the first place.
Oh, come on. One bad decision just makes it easier to make the next bad decision. I mean,
Like
isn't
the
you just
only
keep doing, just go down the rabbit hole, Justin. Come on.
like the only way, like the first idea, at least you're getting a secondary product out of it in some way, that you were creating a potential revenue source that drives the thing forward without you. This one all looks like expense.
Potentially, it depends on how it's implemented. These are just concepts, these are categories, and they are not the only ones, but these are some of the major places that
Yeah.
people are looking. So
Oh,
who
okay. I'm
knows?
not defending all attempts to fix
The
climate. This
can't-
isn't what I'm talking.
Everything is going to be with expense. But can
Well,
we
yeah, but
create it, at parity? Is that possible? Anyway, the take home is there are people looking at solutions out there. People are talking about solutions. Researchers are making leaps and bounds and strides, and solutions are there. It's just whether they're the right solutions and whether we can work together to put the right ones into place to actually put our climate on the right track. You want to talk about something not climate?
Yeah, we can
Sure,
switch
maybe.
gears. Yeah.
Let's talk about the universe instead.
So the standard model of how galaxies are formed, we have early universe, and then the Big Bang happened. Oh, I guess that happened first. Then we have the early universe, and what we would expect to see if dark matter was involved in the early universe is when we look way, 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 way back with the TWIS capabilities, we would see some of these earliest sort of star formations beginning to clump together. But that's not what we're seeing. Instead, the oldest galaxies we've seen, right up into the, some tens or hundreds of millions of years just after the Big Bang, are way bigger than we thought they should be, according to the cold lambda cold dark matter theory. So we're seeing formations of galaxies that are big and bright and much more kind of put together than we would expect. And so while this is not in line with dark matter predictions, we weren't even supposed to see, I think, 1 billion years into the formation of the universe, where we're supposed to be seeing these large galaxies and brightness of stars that are coming from them. But it is in agreement with an alternate theory of gravity, according to new research from Case Western Reserve University, published in Astrophysical Journal, the Astrophysical Journal. What the theory of dark matter predicted is not what we see. This is Stacy McGaugh, Case Western Reserve astrophysicist, who's written a paper describing the structural formations of the early universe, I think under this Aaaand theory. He says, this was actually predicted. This is what we would expect to see based on predictions made back in 1998 under modified Newtonian dynamics, or Aaaand, which is an alternate theory of gravity that has problems. It's not like it's the better version. It has its own.
That's what I was going to say. You said Aaaand, and I'm like, wait a minute. Isn't this one of the ones that is not highly accepted as
correct,
as an option? Okay. Yeah.
So it's got apparently it's got some problems.
Yeah.
However, if there isn't one that is right, like, like there's not a description of gravity that's working everywhere. Like the classic examples, like Newtonian gravity, works perfect. We can land on the moon with Newtonian gravity alone, and then But then it doesn't explain any of the effects we see with Einstein and time dilation and any of this stuff. And so it's not the complete theory. Following standard model into lambda, cold dark matter theories, and then you have an expectation of what the large scale universe looks like very early in the development of galaxies. And Mond says, no, that's closer to our modified Newtonian. So it will just take some time. All these things will get worked out. But I think what sort of attracted me to this story was actually the ending. This is again Stacey McGaugh, and he's saying the bottom line is I told you so.
Yay, scientist. Okay.
I was right. I was raised to think that saying that was rude. But the whole point of the scientific method, make predictions and then check which come true.
Yeah.
So this is the prediction from Mond that you would see very fast assemblies. So under Mond, the idea would be after the expansion. Well, I'll start with the dark matter. Dark matter was you have the Big Bang, and then dark matter is slowly pulling everything together to form galaxies. And that's how you get the spaces in between where galaxies are and the sort of networks and the webbing. Mond says, no, it's there is no dark matter. It's the gravity within those the things that have expanded. And just as the universe expands, it expands with them to a certain point, then gravity takes over and everything collapses. There's nothing.
Yeah. So, but there's no in between, right? So what we need is something that kind of maybe can bring different ideas from different hypotheses.
Right. Well, in Mond, all this will happen right after the initial expansion. Right. And so you'll get galactic formations right away. In dark matter version, it would take a really long time because the dark matter would be clumping things together slowly. Anyway, right now, Mond is winning this prediction.
This particular prediction. This one prediction.
Right, very specific prediction.
Yeah.
Yeah. So again, this is what's so fun about gravity is it seems to be tantalizingly meeting everyone's predictions and not in many places and then not in others.
So the challenge will be that for the person who's able to find the pattern, to see, the things where the commonalities and the differences all overlap. What's that Venn diagram? What is that theory of the universe's formation or the theory of gravity, right?
Yeah.
So they looked at really cool stuff to figure this out.
Well, it's JWIST. So JWIST is looking back further than we've ever seen before. There's another study out too where they found like way back in the early through JWIST these what look like red galaxies, and they look red through JWIST's eyes because they are full of dust, and so they sort of have this big glow about them. It's a separate study from the one we're talking about here. Mm-hmm. But those aren't supposed to be there. Those are too massive and too bright and producing stars. They say these are like Milky Way sized galaxies.
But when we talked with Ethan Siegel, he said Milky Way. Right. He said it wasn't quite as evolved as our Milky Ways, but it was that size and there was star production. And so there was.
Right. Well, he also referred them as proto galaxies, which is fine. Like we're still talking about the early universe. The thing that Mond is also would have been predicting that the current standard model of the cold dark matter lambda CDM is missing is that the star formation is much higher than would have been expected, which kind of again fits with the Mond and not with the CDM. But because these Milky Way sized galaxies are producing stars at extreme rates that we wouldn't see from a current galaxy. We wouldn't expect to see from a current galaxy that size. So, yeah, and this is a very exciting time for cosmology. There's going to be lots of interesting debates and new discoveries being made by this. But more and more, it looks like dark matter is almost not present in the early stages of our universe.
Which is fascinating. So it's still, what is it?
Or it could be nothing, right?
It could be nothing. But what
are we seeing that we're interpreting as matter? And the answer is we're interpreting gravity as requiring
it's mass, right? It's mass and mass and gravity. So concepts.
And there's another one where they're looking for dark matter, and they're finding that, of course, the closer we have gotten to the ranges where the places we haven't looked, and the ranges that we're looking for them, the more we're running into something called neutrino fog. Mm-hmm. And there was another study out this past week where a lot of the dark matter detectors worldwide are like already hitting neutrino fog, which means if it's below this threshold, all we're going to get is noise. We're not going to know if we're finding
unless we get better detectors.
Better detectors are going to be tough
until you can figure out what the noise is to remove it. Right.
The problem is what you basically have is this atom getting interacted with by something on the outside? Mm-hmm. And neutrinos, to a certain extent, are going to continue to be doing that. And that's how they were looking for dark matter. So they're going to have to find it on the cosmic scale in a different way.
Our detectors are going to be galactically big. They're moving that direction already.
Yeah. I
love it. We're going to find all the things. We're going to figure it all out, at least in the next 2 years. Of course, it's super easy. These decades researching stuff. Who does that anymore? We've all wanted one ring to rule them all. What about one genomic test that you could take to find out what's infecting you? Oh my gosh, if you didn't have to go to the doctor and, let's take a flu test, let's take a COVID test. Oh, you have to take 2 different tests. Let's take a bacterial test. What? We don't know what it is. Take all the tests and they've
lab on a chip. Is that what you're talking about?
It's kind of like that. Yeah. So researchers at UCSF have developed what they call a next generation metagenomic sequencing test. And they say that it has proved very effective at quickly diagnosing almost, I don't know what almost is. I mean, really, truly, what is that? Any kind of pathogen. So what they are using is this NGS, metagenomic next generation sequencing. It speeds up sequencing time, analyzes not just DNA, but nucleic acids and RNA, anything that's in a sample. And the researcher, Charles Chu, who is a professor of laboratory medicine and infectious diseases at UCSF, says by replacing multiple tests with a single test, we can take the lengthy guesswork out of diagnosing and treating infections. So it's kind of like lab on a chip. They've performed this test on thousands of patients already. So they have used it. They have developed it already to analyze cerebrospinal fluid. So this is very specific fluid, babes, the nervous system.
Wait, yeah, that's not something you can just do at home.
Right. No, this is not spitting in a test tube or going and just getting a blood test. CSF is, you have to get a spinal tap for them to get your cerebrospinal fluid, or there has to be a hole in your head. And these patients who were tested, they had unexplained neurological symptoms. They've now published a paper in Nature Medicine demonstrating that their test on these people with neurological conditions, they correctly identified the pathogen 86% of the time. And then they had a different study in Nature Communications, and they identified respiratory fluid. So this is from your lungs. This is stuff you're breathing.
That's
This
a little
is
easier.
exhaled, inhaled. Yes,
Yeah.
this is stuff that can cause pneumonia. And they were also able to identify pathogens in respiratory fluid with a very high success rate. So the researchers say between 2016 and 2023, there were nearly 5,000 CSF samples. 14.4 of them actually had an infection, and then 86% of them, of those 14% that were actually infected and not something that was just physically wrong with the individual. They were able, 86% of the time of those 14.4% to actually identify the pathogen. This is better than any other category of test for neurologic infections at this point in time. They worked it up to be able to also do the respiratory stuff. And their goal was to have the entire process completed within 12 to 24 hours. So kind of like the COVID tests that we started taking at the beginning of the COVID pandemic. They adapted it to work with respiratory fluid. And the CSF test takes 2 to 7 days to process, but the respiratory can take 30 minutes of hands-on time, and then robots and algorithms can take over. And so it can be less than a day before they can detect viruses like SARS-CoV-2, influenza A, B, RSV, and many others. So I'm excited for the future. I feel like the testing is going to get better. I'm going to do great on tests as I get older. Test prep has never been easier.
That's pretty wild.
I mean, number one, the lab on the chip idea, we know that there is a whole company and a movie based on that company, or a TV show or whatever. There's the lab on the chip idea, being able to submit a drop of blood or a sample of saliva and have a lab test tell you exactly what's wrong with you. We are, as much as our technology is really, really good and accurate, the efficiency, and like we were talking about with carbon dioxide, and the scaling of testing is just not to that point yet. And it was taken advantage of by a company and people in the company who wanted to make money. And so I hope that lab on a chip kind of ideas don't end up being delayed in how they're brought out to people in the future because of the greed of people in the past. Anyway, this I think is incredibly, it's going to be beneficial, and I'm very optimistic about it on many levels. Yeah.
So
breathe easy. Breathe easy. Yeah, breathe easy.
So I'm actually surprised that we don't have lab on a chip. So I can see why that scam worked.
I was talking with somebody decades ago, I mean, probably 15 years ago, about in a lab. No, ages ago, like definitely they have been looking at proteome and genome and all these things. Like these things they're there, but just the scaling hasn't been capable. Like we're not there for whatever reason.
So I think we might be, though. I really do. And that's why I've started a new company that's doing it.
Justin's Lab on a Chip. Dorito flavored. I mean,
isn't that how this works? Yeah, isn't that a problem? So this is, again, I'm going to throw out a couple of stories. There is Massifold, which has taken protein structure predictions from months down to hours.
Absolutely. Yep.
And this just got released as an open source version. Illumina recently came out with a new gene analyzer that can accurately detect single nucleotide gene variants and match them up with genetic trait diseases and all sorts of and it can do this, I think, within minutes, if not an hour at the most. Or actually it can do, my goodness, it can do hundreds of these in an hour, not a problem, with whole genomes.
Yes.
Single one probably in just minutes. But when we're talking about those thresholds, the scalability aspects, basically what we're talking about is computing strength. And as we've gotten into this world of artificial intelligence and machine learning, and that thing is becoming now at the fingertips of anyone, the real capabilities for analyzing the data I think part of it is you need something that's going to collect your data for your lab on a chip, right? That part is, trickier than we might think on some level. But if we took it down to a genetic level, right? So now we have a lab on a chip, which, by the way, I'm trying to look for something that's the same size of the
lab on the chip that you're using at work.
So the Oxford Nanopore, which
and I really love the auditory aspect of this, Justin. I mean, our podcast audience is gonna love
this. This is a charging thing for a laptop.
For your laptop.
But this is the size of a modern day sequencer.
How what is that? 2 inches by 6?
Yeah.
By
This is a
2 by 2 by 6.
Call it the size of a candy bar, and you can put material in, and it will read the genome of whatever's in there, right? And so we have that. So then you combine something like that with the ability to analyze it in real time, within minutes, half an hour, put it on the thing. It's surprising to me that we aren't there. The databases of what is a pathogen's genetics. So how can you not identify it from something like this, right? All the pieces are there. And that's why you should send money to this show. If we raise, we don't need a lot. $500 million should be good seed money. We'll have this product out within probably a couple of weeks, I would assume.
For sure. I'm
all
sure
the components.
You could use the night staff at whatever genomics lab.
all the components seem to already be there.
Yeah. And there are companies that are aware of all of this. And there are companies that are also capitalizing on the fact that there is cloud computing power, that we have the ability to quickly get information up into the cloud, process it using massive amounts of servers, similar to all the AI processing, and also using large data models for genetics, proteomes, all of these things to be able to identify this stuff. Like you said, the pieces are there, and more companies that provide remote lab facilities, that provide remote lab analytical capabilities, these companies are going to be, used and really on the forefront of where we're going.
Oh, I thought you were about to say they're going to be gone.
No, I think that they are the ones that they're going to be the interface, right? They're going to create that interface between the person in the lab who wants to do this thing and, you've got a large data set, there's a model, and somebody creates the interface to make it easy. Mm-hmm. Somebody also has to maintain the processing server time. Right. So all that stuff has to be paid for somewhere, and no one university or research lab is going to do that. It's going to be.
This is what I'm saying is that's what's also been changing. The massive fold you can run on a laptop. It's a deep, this was a massive servers, AlphaFold, and huge amounts of computing time and energy and resources.
Right. They're using graphics cards, and they're using now
you can run it on your laptop. The sequencers used to be these, one thing is running the sequencing machine, but now it goes to the servers and the processing and everything else. Now you can run that on a laptop.
I feel like there's going to be a combination of things. So there's going to be you can download a certain model data set or a model that contains whatever that knowledge is, but it's not the whole data set, right? And so you're kind of stuck if you're out in a remote space, right? Say you have to go to, the Congo and, try and figure out what infectious diseases are emerging there, right? So you have a laptop, which will work because you got this heavy duty laptop that's waterproof and all the stuff.
You've got the candy bar sequencer fits in your pocket.
That you got your little Q-tip swabs and little things to do blood and spit and all the other stuff, monkey feces and whatever it happens to be. I mean, welcome to South Carolina, I think it is. I don't know. Atlanta, Georgia. I'm not sure. There's a few monkeys not back at home yet. Anyway, monkeys. But it's going to be this combination of the places where the data, the models are built, updated, housed, where researchers are continually updating with new genetic information, new understanding. There has to be that accessible compendium, but you also have to be able to take, whatever it is that's downloaded to a computer to be able to attach to your little chocolate bar and, do some on the fly stuff, but it's never going to be quite as accurate or as immediately to the global scale of understanding as being remote, being able to go up to the cloud and access the power of the cloud processing and what's going on there.
But again, this is what
I'm not saying you're wrong. I'm saying there are different pieces to the puzzle and there are different ways it's all going to work together. But
yeah. What I'm trying to say though is that's what's changing. That's it's been this last couple of weeks. There have been a few just absolutely game-changing technologies that have been put out that have shown oh gosh, even we were talking about this maybe 5 years ago, but looking for single nucleotide variant changes and gene variants and stuff would take a team of bioinfo magicians staring at their computers weeks on end searching. Is that a signal or is that a miss? And now this, now they've got gene analyzers that are doing these things in minutes. It's
we saw it coming and we've seen the prices come down and we've seen the time come down and now it's, but it's still, it's not perfect. It's still growing. But it's changing and it is game changing, like you said.
But for like I said, for $500 million, I'm sure we could get this thing off the ground. It's not.
Come on, everyone. We'll both be the face of the company.
I think it won't be me. I have an insincere affect. I would never get away with. Even as an honest broker. See, even the way I talk, it sounds like.
I don't believe that, but I mean,
even if I'm being honest, nobody would believe me.
Well, I'm no Elizabeth Holmes. That's a good way to. I don't lie. I'm not good at it. I can't do it. Oh my goodness, we have so many more stories. Let's see. What do we have next? You've been talking about. We've been talking about this one test to rule. What about fixing stem cells or vision with stem cells?
This is the research from Osaka University in Japan. They have conducted the world's first human trial using induced pluripotent stem cells to restore vision. It's only 4 subjects, very small sample size, but that is kind of what you'd expect from a first ever human trial. So actually maybe 4 people at once is a very large first trial.
Ish.
So
large-ish. Yes, exactly. It's not 1 person. It's but 4.
Usually when you're talking about a first medical procedure on humans, Like, oh, we put the one heart in from this kind of new kind of a heart, or derived from and grown in an animal lab or a mechanical heart, into a human to see if it would work. We don't do it for, we put it into 4 people to see if it worked. Like, you don't, typically do that.
It's better than 4 sheep
But
for human work. Yes.
yeah, so these are folks who have a condition where they have lost function or availability of adult stem cells in their eyes that do normal eye repair. So your eyes aren't just, the lenses that you have from birth and things have been unaltered. You have adult stem cells going in there and making micro repairs all the time. And if they stop working, you start to get these fibrotic conjunctive tissues over the cornea, which can reduce your vision or even lead to blindness.
But that's not the same as glaucoma.
I don't know. I don't know what causes glaucoma. You should talk to your doctor. So
Sunlight. I don't know. Sugar.
You should ask a doctor.
Yes.
But not a dentist, because they're not real doctors. They don't know anything about it.
Going back a couple episodes. Okay.
Yeah, I know. Still. So these are limbal stem cells that are normally performing these repair functions. So they become compromised. You get fibrotic tissue buildup, ultimately vision loss. And traditional treatments are going to involve grafts, where you take a section from a healthy eye. If your other eye is not having this, maybe switch it over, take out a little patch, put in this patch. So in this one, what they did was they had these pluripotent stem cells that they developed in a way that mimics how normally they would develop in the eye, so that they were sort of creating all of the necessary pieces that would be used in the limbal stem cells. And they did these transplants into the 4 individuals. They monitored them for 2 years, and they found that, all 4 of them had improvements in vision. One of them regressed. They also say, though, that this person had the most aggressive, most aggressive or advanced, I guess, problems in the outset, but they had all progressed to getting better vision. Of course, improves quality of life and everything else. So, of course, the next step is to do a larger trial. Try it again.
More than 4 people.
Yeah. More than 4. Now it will need to be multi-site. It'll need to be replicated. It'll have to be a larger sample size.
So I think this is interesting because we've already seen some success with gene therapy in certain types of eye disease, where they've gone in because of certain single nucleotide polymorphism things that they've been able to address, and actually apply gene modification therapies to them. They've been able to show vision coming back to people, but they're not all problems can be solved with just that simple of a solution. And so the issues we've seen historically with stem cell therapies is that the stem cells come in, either we didn't understand specifically that they had to be kind of a certain tissue type or at a certain state, like ready to work in the way that a usual stem cell would. It was more of this like stem cells are going to fix stuff. And so it was, unfortunately, it was naive at the time, but it taught us a lot about what we didn't know, to the detriment of people who were unfortunately already suffering from terrible diseases. But I think it's the way that we've advanced with this technology to be able to apply it in a way that is biologically appropriate to fix a problem. I'm excited about that, and I want to see how it can go on and help, and if it is a lasting solution.
Yeah, They also had a sort of secondary experiment within their small sample size where half the patients, were given organ rejection drugs, or a drug that usually prevents organ rejection. And they did find, although they saw improvements on all of them, they did find a better response in the folks that had that. So even though that was, they didn't think that they were going to encounter that, they kind of added that to this round of trial and preclinical trial. So now they've even learned a little bit more about, even in the instance of a pluripotent cell that's designed to be this cell tissue, you still need a little bit of anti-organ
rejection.
drug.
Right. The immune system needs to go, you're same. You're not other. Yeah. I accept you. I will not fight you off. Yeah. And I think, that's another part of it too. Like we've talked about other stem cell therapies where it's like, let's just make big batches of stem cells, like blood donation, right? Where let's make big batches of stem cells that anybody can use, right? That we can just give anybody who needs this the right pluripotent stem cells. Doesn't matter where they're from. We want to be able to give them to people. And the idea is that the pluripotency of them, the fact that they're early, the body shouldn't reject them, but there must be signals. There must be some kind of markers that the body does respond to. And our body doesn't want other stuff in it.
Yeah. And there are universal lines that are being developed that,
of course,
that are avoiding rejection on their own. And these ones weren't rejected, but it did seem like
it improved.
It did seem like it was they found an improvement there. So
yeah,
more to unwind and to figure out.
I like unwinding, unspooling. Let's just let the good times roll, everybody. Yes. This is This Week in Science. Thank you so much for joining us this week. We are talking about all sorts of great science news stories. And if you are enjoying the show, please head over to TWIS.org. Check out our show notes. You can also see a couple of places to subscribe to This Week in Science. But if you're not finding what you need there, just know that you can look for us all over the internet at TWIS.org, our website, or This Week in Science, or at TWIS Science is where our usual social handles are. And if you head to TWIS.org, that's also where you can find links to support the show. So if you enjoy what's going on here and you'd like to throw a little support our way, our Patreon link is there. So you can join Patreon for TWIS and become one of our monthly or annual supporters. You can also click on the Zazzle link and go buy TWIS merchandise, and that'll support us as well. We do appreciate your ongoing support in order to maintain the basic nature of this show. We really can't do it without you. Thank you for your support. All right. As everyone is busy sharing and liking the show, I'm going to come right back on in and bring Justin back in because, you know what? I think Justin has more stuff to talk about. Don't you?
Oh, maybe.
Oh, perhaps.
I guess this might be is this my? No, there's a couple more stories.
Your mic switched when you left your desk during the break.
Did it really? Oh gosh.
Yeah, okay. Rachel, this is a cut and a hack.
Yeah, why does it do this? Ugh.
Because convenience. So, computers know what you want more than you do. Anyway.
End cut. So there's something very interesting on the ceiling of the Sistine Chapel. Well, some people would say there's a lot of interesting things. And if Kiki, if you could share the image that is involved with this, so I could, maybe I can do it.
I got it. I'm bringing it up.
Yeah. Okay. I don't know if you can zoom from how you've got that there. But if we look at image 1A, here's the first one there, and we zoom in on the mother.
I'd have to download it. Okay, give me a moment. But just go ahead and talk, and I will see what I can do. I didn't realize you needed to zoom.
Yeah, we're gonna need to talk about this one a little bit. This one's
Okay.
a little okay. So what we're looking at here is Michelangelo's The Flood, or sometimes called The Deluge, which is on the ceiling of the Sistine Chapel. The study here was led by a forensic pathologist and a group of experts who were looking into this fresco because the woman in sort of the lower left foreground appears to have breast cancer. Now, for those who can't see this, you should pull up The Flood or The Deluge while listening here so you can also see this image. This is a depiction of the Noah's flood. A guy got angry, upset about something. I don't know what it was, but decided we're going to start all over. And you can see in the distance, there's the ark sailing away as the world floods and people are struggling to get out of the water.
Sorry, y'all. You didn't make it.
Yeah. And so this is part of something called iconodiagnosis. It's a very real process of medical analysis that looks for clinical signs of medical disorders and diseases in artwork. It's often used to provide information on pathologies in different sort of paleo-historical periods where artists have captured these anomalies, but maybe medical science had or had not diagnosed these things at the time. And so we can see now we want to move. You've got a nice image there now. Can we move it down to the lower left? Okay, there we go. So, and actually, if you go all the way down just for a moment, we can see what we're talking about here. And so we can see the breasts. There is a nipple that is sort of what do you call it?
It's malformed. It's
malformed. It's
intruding as opposed to extruding.
Yes. Yeah. And there's some lumps that are appearing top, and then we'll go back and up to the mother and child there again. Now this image here. Yes. Perfect. Right there. Right there is perfect. And so, yeah, they looked at this and they come to the diagnosis that it is presenting as breast cancer. And they kind of attribute to this Michelangelo having decided to do this partly with his familiarity with human anatomy. They point to the fact that Michelangelo spent a lot of time doing autopsies and dissections starting at the age of 17, where he would have likely observed this pathology and others. They finding these very specific pathological abnormalities in the woman's right breast might then suggest that this is an intentional portrayal of the disease, not just a theoretical one. Some of the things they're up against is, gosh, this woman seems like way too young to be getting breast cancer. So is it?
How do we know? I mean, it's not. Yeah, that's what
It's a painting.
But we know that, and she's wearing a cloth over her head. She's got a child, but
she's got a cloth over her head, which would indicate that she is married.
Okay.
Right.
Not that she's undergoing chemotherapy.
No, because this was a long time ago.
Yeah, exactly.
Right, and so then they're like, with comparisons of other works of Michelangelo, it's of course. He's, of course, quite capable of depicting a healthy female breast. There has been previous papers on something called Night in the Medici Chapel, where there's a breast there that seems to be portrayed with some abnormal symmetry. Not as pronounced as the one they've discovered here in The Flood. It would not have been unknown in Renaissance times. In fact, the Renaissance is kind of renowned for it sort of being the first golden age of of medicine and anatomy, where these things started to become sciences.
Right.
So the research analysis concluded that the features observed align with the required evidence for an iconodiagnosis consistent with breast carcinoma. They go on to say the theoretical context of the flood illustrates a divine punishment being handed down to all humanity. And so some of the characters in we need the image still. We're halfway there. You can see people on the boat are fighting each other. People have identified different sins that mankind is continuing to carry out during this. Here's the thing. That woman, it doesn't make any sense. She'd be suffering from breast cancer, whether God was flooding the world or not. We need to go back down to the image there. I'm sorry. Go back down just go down to that lower left there. Let's go over here. Oh, the other way. Yeah. Okay. And we'll go up just slightly. Just the other way. No, not this one. Not this one. Up. Okay. So that mother and child almost seem to be in their own world.
They do. Yeah. She's sitting on there's a cut tree. There's a rock maybe that she's against. She's got not just the cloth on her head, but the cloth is wrapped around her abdomen in a way. She's resting backwards. Her child has what appears to be a bandage around its head. Could just be
is holding something in their hand. Can you see that? So
yeah.
So then past this story, here's what I think this is. This isn't part of the study anymore. Here's what I think this is. I think this is Michelangelo. I think that's
his mother.
Yeah. So when I first saw this, I thought this
fascinating
was Michelangelo.
But there are many children and their parents. There are children holding on to there's a strong woman above who is holding a child, one child clutching her leg. Everybody is in the nude or barely wrapped in cloths, which is exposing the humanity of ourselves, right? But you're right. The lower left here, it's individual, unique. So it's not the masses, right?
Right. And the fact that she's suffering from breast cancer on top of the flood, it doesn't seem like an additional punishment. They don't seem to be engaged in any kind of sinful activity or any indication that they would have been involved in. Anyway. So I
want you to talk to somebody. Is this Michelangelo? Is this
subversive? This is what I thought. Is this some sort of subversive thing? That Michelangelo, having done all these autopsies, is like, yes, you flooded the world, but you've also been afflicting women, God, all this time. But and so I started looking at Michelangelo a little bit, and he does not seem to have an ounce of, disbelief or lack of faith or fall from faith that I could see. It doesn't seem to be that. There's a horse that seems to be looking over them. There's the ark in the background. Was the horse late?
Right.
Why is there? It's the only animal in the
horse
picture. Is this horse, and he's just.
They already have
two. Looking over
they already have two horses on the ark. I mean, come on.
So, then what I found out about Michelangelo's story was really interesting. Michelangelo was sent to go work to go live with some stonemason people, some stone workers, in one of the mines owned by his father. Quarry, where they were pulling white marble, which is actually on the other side of the image, where he learned his love of
sculpture.
Learned his love of marble and started his career as an artist based on these things.
One of the greatest sculptors.
So I was like, well, why was he sent there at age 6? Well, because his mother died when he was 6. Which is about the age of this child. What did his mother die from? Well, his mother died at age 30, of what is only described in anything that I could find as a prolonged illness. What is the other interesting thing is the only other story that you will ever find about Michelangelo's mother is that she was pregnant with him while riding a horse, was thrown from the horse, and they both survived. And within the family, it was considered a miracle. It happened to happen on St. Michael's Day, who was an archangel, which is why they named the child Michelangelo. So here we have, a woman who's too young, seemingly, to have cancer. It would have been a rare event with the breast cancer. We have her about the same age as Michelangelo's mother when she died. We have a child that's about the same age as Michelangelo. And we have a horse that is looking over them in this condition. And she almost looks like she could have been thrown from that horse. Like she's in almost a fallen position, not a normal resting position, the way she's laying across the log. And then all of these probabilities led me to think that that's Michelangelo. But then he's holding something in his hand, that child.
Okay.
I think it could be.
I'm listening to your story. Where are you going?
It could be a chisel. It could be a scalpel. It could be that this is in the flood of the world. This child is already crying for his mother, who doesn't seem to be paying attention to the flood, but seems to be crying for his mother, who's still there. This may be Michelangelo grieving for his mother in the theme of the world flooding.
And we also see his mother not caring for him, but concerned over her own disfigured breast. She is looking down at herself and not he. But
the child is on her shoulder, right with her.
Right, but there's it shows a different care than the other mothers in the scene.
Here's the last
which
piece
is interesting. Yeah.
Here's the last piece I would put. What hand is the child holding whether it's a chisel or a scalpel? 10% of the population is left-handed. What was
Michelangelo? Left-handed.
Final piece of the evidence, bringing it all together.
Look at you.
Yeah.
And this is only one small part of that entire ceiling.
Yeah.
This is just one little tiny bit of it.
But I think it adds to like, why did he deliberately portray with such specificity breast cancer in this image? And it may be that, he is learning in life, one, that breast cancer, what that is, and then maybe hearing from father or family that, yes, your mother died from an affliction of the breast is what had taken her, as he got old enough to know, right? And so him putting it together and then representing it here, I think is logical. And his slight obsession with it, if he has portrayed it in earlier works and sculpture.
Yeah.
Yeah, I think that might be a little Michelangelo in the background there, self-portrait.
So there's multiple levels of inquiry, right? The scientific inquiry into the historical relevance of the works of this particular artist, which have been seen by so many millions of people.
Yeah,
millions of people.
Yeah.
Have billions probably have, like, over time visited since this was painted, to visit and see the beauty of this Sistine Chapel. But how many have looked upon it and delved into these very unique aspects of the story that Michelangelo is telling?
And if you scooch over to the right,
I know there's a dude carrying like a thing, and he's this bald dude. I know. But
I don't know if you know. That's not what I'm talking about.
I know. But what I'm curious about. There are the babies who have and people who have very anonymous faces, and then there are some individuals who are looking off in different directions in like really specific ways. It's one bald dude who's just like carrying an upside down table with pottery on it. He's I don't know. This is fascinating. Okay, all the way to the right. Scooch into the right.
And then up. Oh wait, I'm sorry. It's hidden by these extra images on this one. Well, right below this little shelter here, you see with the tree, is
I
an
don't know. The
outcropping.
podcasts are not going to know what's going on. So there's
But
an
if you're
outcrop.
looking at the flood or the deluge, it's hidden by
Yes.
the image we've got up there now. But
Yes.
there is a big outcropping of white marble hiding behind that overlaid image that they have there, with the only other shelter above ground, which is where Michelangelo then gets sent to. I think he's telling his story, but I think I'm very curious with that. Knowing that he was a very devout person, it doesn't look like it is
Yeah.
a. It looks like he was in a conversation, and he's
Well, I mean,
running through the details
several
of.
years. He told a story that took him years to present.
Yeah.
But how many other people did he present? I mean, as we know, like you mentioned, him studying as an anatomist, how many other people did he present in like medical ways? How many clinical kind of representations
Yeah,
are presently
apparently
found?
not very many.
Mhm.
Not very many. When he has the discussion is usually around breast cancer.
Hmm.
He's at least one other sort of verified time he's done that, and Night in the Medici. So yeah.
This is fascinating. I love from this study that you found in the journal The Breast, sentence in here of interest, the BRCA 1C.4096 plus 1G greater than A and the BRCA1 1499 insular A GPV have been identified as founder mutations from 1800 years ago in the Tuscany region and subsequently disseminated. And they are rapidly associated, increasing breast cancer at a very young age. So it's an interesting, just because there was less life expectancy during the Renaissance, even though medicine was
So
coming, this was
yeah.
all these factors are so fascinating together.
And the thing about life expectancy is it's a very big misnomer. Michelangelo lived to 89, something like this.
Not
So
everybody did, though.
no, but I mean, life expectancy also was a lot of times the reason it's like, I think it was 35 or 40 or something like that around then. But it also meant that a lot of children died.
Yeah.
Like the infant and child mortality rates and childbirth deaths and these things.
But if you are of a BRCA mutation, that it is for younger breast mutational inheritance, and your children live, then you are more likely to pass on this younger. Like you won't die before you have kids. You'll, and also the breast cancer isn't something. The ages of everything kind of work out so that you'll have the breast cancer and
And
then
I
you
didn't even
pass
think
it
of
on to
that.
your kids and maybe they'll make it too.
I didn't even think of that, because now you're telling me, and then I've got to think of it in terms of not just his mother, but maybe there's a family story about, like, yes, this has happened a few times. Like maybe there's aunts that can weigh in, or uncles who.
Right, because there's a difference between the breast cancer that occurs as the impact of age, and that
Yeah.
there are mutations that are definitely longevity related. And like the probabilistic nature of whether or not they get activated to really become cancerous.
Yeah.
And those people, yes, but they're going to have kids that live full lives, that raise kids that live full lives, and the cancer doesn't affect younger populations. And so it's the fact that it's this particular mutational category that even though there was a young child mortality rate, if the people with those young breast cancer mutations were living long enough to have kids and to like raise some of those kids, that would get passed on. And that can, and the fact that it says this is Tuscany, is this region where it manifested, is, I think, really interesting. Thanks, Sistine Chapel. Thanks, Michelangelo. And this is a reason why people need to continue to support the arts. Because who knows what people will capture from this day and age that is important to the interpretation of disease, life, history, culture, all this stuff. Support human arts. Did you have another story now that we've had this really, really fun conversation that's going to take me down for a second?
Yeah. This is actually from Christina Tonshoff over at the Conversation.
I love the Conversation. Great group of people. Yes.
It's just a quick thing here, but representatives of the world's governments are gathered in Baku, Azerbaijan to negotiate the international climate policy at the COP29 summit.
Number one, why Baku? But let's go on from there.
So, well, the last one was in Dubai, which is exactly where you're going to get. So
I know.
Honestly, and this one is going to be mostly, apparently, is going to have a large focus on the finance industry, which is no doubt going to be focused on how to monetize climate change.
But the science, right?
Nope. Finance.
All right. I'm going to mute myself for a while.
So there's going to be expected about 30,000 participants that are there. Maybe 10% of them are for sure lobbyists. What this article is kind of pointing out is that climate lobbying isn't as direct as lobbyists going over and trying to influence any legislation or any wording or drafting of commitments.
So not the same as lobbyists that we would expect, like working within our nation's capital.
So yes, there's going to be that. That's about the 10%. But what they're also pointing out is that a lot of the lobbying happens before anybody gets there. Some of the lobbyists are on the committees. Shell Oil Company, point out, has previously sent representatives there who were working for were sent there by different governments as their representatives. There is a non-governmental committee registry that you can, if you have a group, you can pay and be part of this. These are almost, they think, entirely, almost, not completely, but almost entirely now made up of groups that are lobbyist groups. So it was Brazil.
Lobbyists can be anything. Like, it's not, we say not lobbyists, that lobbyists are trying to get things done for groups with different agendas.
Yes.
Right.
The minority of those.
I'm trying to just make it.
Sure. The minority of those are going to be people who are concerned about climate. The majority at this point are people who are trying to affect any sort of agreements that are made. Again, it was Shell Oil Company had direct input on draft resolutions because they were official country, part of the official country delegations from Brazil and Nigeria in the previous COPs. What it looks like is there are now sponsors, corporate sponsors for these events that then get special access to pre-draft documents. They get to read things in advance by being a sponsor.
And like for journalists, be like, I don't like the way that was written. I don't think it's accurate. Maybe you should reword it before it gets published.
What this all looks like is a carnivalesque grift. It basically just, and the fact that you're holding it in Dubai and Azerbaijan Do they have different laws when it comes to these things? Are you allowed to hand a bag of money? And also, this is the politicians who are going there are coming away with donations back home to their campaigns.
Yeah.
Unrelated, completely unrelated. Maybe, probably not. I don't know. I'm at the point now where I think the UN is just bullpucky. I think this whole COP thing is now just a grift. I think it's become at some point they realized it was monetizable to go to these events and pay lip service.
Everybody knew, they thought they could get away with it at first. I mean, the issue is it's not the UN is grift. It is the people are using the infrastructure and the platform for grift. And these companies believed that earlier in the process, they could market and propagandize their way through it and get what they needed done. But then they realized that public sentiment was changing because the science was coming out more clearly and in different ways. And so they had to be there. And then the argument was made that economics is, everybody votes with their pocketbooks. And so economics needed to be one of, or if not one of, the main lever for changes around the world. And so it became more of, the UN has it has no sharp nails or teeth. It cannot do anything itself, but there are peacekeeping forces, right? With the UN peacekeeping, but it is an organization that brings the world together to talk about stuff, to do things, and hopefully make agreements that everybody will stick to. But who is the regulator? Who's going to keep it going? I mean, we have these issues in our own government. If the FDA, the EPA if we set regulations and laws, but we don't put money in the budget to hire the people to enforce those regulations, suddenly we have E. coli outbreaks because you couldn't have enough people going to look at the lettuce farms. Suddenly you have, issues at, Frosty Freeze little thingies and, whatever.
But I guess my point is, and I totally understand that. I understand that the UN.
So am I on a tangent to what you're saying, or are we on?
No, I think we're in agreement. I know that there have been all kinds of false promises made at these COP meetings before. What I'm now realizing, thanks to this article a little bit, but also from a lot of other past and current information, is that this is now like the annual meeting to combat climate activism, to combat climate information.
Or for everyone to get together and go, look how much we didn't do. Oh my gosh.
And to come out and to meet the people you need to strategize so that they will write your law back home that makes protesting oil pipelines illegal, which it is in the United States now. Do you understand this? It's illegal in the United States.
Yeah, active terrorism. I know. Yeah. Nonprofits, not terrorist organizations yet, which is great.
So stop letting them meet.
The meetings need to happen. It just needs something.
These are not the people. And then their main focus is on how finance can turn a dollar off of this. Yeah. Not at all on science.
And I agree with that. And I think that is to our detriment. And the fact that we have allowed private short-term economic interests to take precedence over global long-term sustainability it's a crime. Like, we should be, prosecuting it on the scale of, war crimes. But since when have we actually prosecuted a war crime? So, whatever.
I keep feeling like I slipped into the wrong universe.
No, you just woke up at 3 a.m.
They just need me here more? Is that what this is?
Same bad dream, different day.
feel like my consciousness belonged in the other one, but okay, fine.
But I do believe in the UN as we need a place for everyone to come together and be
For
able to and peacefully communicate and talk about stuff.
are
I
they
do
involved
not.
in any peace processes right now?
Yes.
Are they? What are they doing?
The,
What do
the
they
UN's
do?
UNESCO is amazing. World Health Organization. There's like amazing stuff that is involved and related to the United Nations.
Is there?
There's yes. Don't boil it down to like a one issue voting thingy.
It's a global level.
I know. Yes. But yes, I agree. The issue is that it has been co-opted by the greenwashers and the people who are trying to figure out how to maintain the dominance of fossil fuels, or at least
Just polluting.
um,
It's polluters in general. It's not just
I don't
fossil
even
fuels.
think that it's not that they want to pollute, it's that they want to make money. So it is, how can we take and not give back? How can we, how does the science end up on our side? It's not even, we want to pollute kind of question. It's not a moral, ethical, like environmental issue. It's not evil in that sense. It is short-sighted and just based on economics, which is unfortunate because that's not sustainable. If anybody ever studied ecology, you would know that that doesn't work out in the long term. So, yeah. And this is why, none of the agreements are upheld. And as far as the reporting goes, at this point in time, no country has upheld any of their agreements for reducing carbon dioxide emissions. Even though there have been reductions in certain areas and not in others, we are not on track and nobody's doing what they're supposed to be. And I don't know. I think it was somebody who Trump would like who said, "Hey, stay in the Paris Agreement. It would be bad for you if you left." So that one I think is a very interesting story. There are undertones and subplots to all of this stuff, everyone. Look for the subplots in everything. Oh, and if you're not looking for the subplots, you must be one of the bored people.
What do you
A
mean
new
bored people?
the bored people. This is an open access study published in Communication Psychology titled "People Are Increasingly Bored in Our Digital Age."
Yeah.
Right? How many times? I mean, kids forever. I'm so bored. But then you go, take care of it. Go entertain yourself. Figure it out. There never will be another Gen X. I swear. For good or for bad, for better or for worse. So in this study, what they look at is reports of boredom, where we have entertainment at our fingertips all the time. You would assume that people can't be bored because we could always be entertained. However, chronic boredom is rising. This can undermine well-being, learning, and behavior. And so these researchers delved into this and they explored one possible reason for this boredom, which is that digital media use makes people more bored. This is through dividing attention, elevating desired level of engagement, reducing sense of meaning. So it's like less hedonic meaning. What? Heightening opportunity costs, and also not being effective as a boredom coping strategy.
Makes sense.
It does make sense. And where we're sitting here right now, we are part of digital media. However, I hope that anybody who's hanging out with us is, listening and thinking and engaging with us because of the way that we talk about things I mean, at times, of course, we're talking for a couple of hours here, right? So at times your mind's going to wander. You're going to go off on different tangents, but maybe we sent you there. Maybe you started thinking about something and you'll come back around. Maybe we are a part of hopefully helping you craft a less bored existence because you are more curious about your world. Once upon a time, my grandmother said that bored people are boring people, and I do not believe that anyone who is a part of our audience could ever be boring. No. There's no bored people here. No boring people here. You might be bored for a moment, but you'll find something that excites you. And I think that's true for life. And the reason I wanted to bring this study is because I think it is very important as we're watching our kids and we're looking at our own mental health decline and people doom scroll. Doom scroll is a word.
It's 2 words.
No, it's like hyphenated. It's like a single word. It's a compound noun, compound verb. Anyway. Yeah. Young people from 2009 to 2020, as they've done representative surveys, meta-analyses of birth cohort changes, everyone is getting more bored.
Mm-hmm.
And maybe we don't have to be. Maybe there are ways that we could reduce our boredom. And the one thing I was talking with my husband about today, Marshall, towel boy, for those of you who have been towel man. I guess he's a towel man, if you'll remember from years back in TWIS history. That's one to go back to. I was talking about the fact that it's just so easy to go online, to get on your phone, to stay at home. You feel like you're connecting, and we have all connected with so many people online, and it makes that so much better, but in many ways. But we're in a point where if you can and you are able, or if there are people you can connect with who can bring it to you, human contact, the human experience, getting a perspective from another person, because people are like random commentary generators, way better than AI. They can bring new interest and new framing to your life. And so we got to keep our connections, everyone. I don't believe it's just digital media. I think it's the way that we use it. And it's also the way that we engage with people. Are you scrolling? Are you thinking? Are you engaging? Why are you doing what you're doing? Are you making the lazy choice? Are you making the one that's actually going to take a little bit more energy because you have to put pants on?
Oh, in that case, I'm out. Yeah, you just crossed the threshold, Kiki.
I know, it's the pants line.
Yeah.
It used to be a laundry.
Once in my sweat shorts.
Yeah.
That's it. I'm not putting on pants.
I'm out. I'm over. I'm done.
It's too late.
Too late for that. Okay, so moving on from being overly bored, if you're depressed and you are chronically depressed in a way that you perhaps have been told that ketamine therapy might be something that can help you out. We've talked a bit about how ketamine therapy is useful for very rapid help to alleviate depression symptoms in order to and actually has ongoing long-lasting effects from single dose therapeutic sessions. Researchers just published their study from the University of Buffalo, trying to figure out what exactly is going on there. Because, okay, we've known for a really long time that ketamine binds to a particular receptor that I was interested in graduate school because it's related to long-term memories. And so this NMDA receptor, the NMDA receptor is usually thought to like kick in as part of like neuronal stimulation. It gets turned on and then connection pathways fire, and everything's great. But it's always been this kind of like, okay, so ketamine gets in there, turns on NMDA receptors, and that should be great. However, the reasons this happened, like how things work mechanistically as ketamine being an antidepressive, has not really been well understood. So if you give ketamine at very high quantities, you get the dissociative effects, you get the sedative effects, you get the effects that, that make people go, I don't feel my body anymore, right? So how does that lead back to you're not going to be depressed? When people talk about ketamine as horse tranquilizer or a veterinary drug, it has been used in anesthesia and is still used in anesthesia to this day because of the way that it dissociates or turns down turn or changes the way that the neurons talk to each other in a way that leads to these impacts. But what they have seen is a dose response effect, so that the therapy that's really, really effective for depression is low dose ketamine therapy. So the research that is just published this last week in Molecular Psychiatry shows, and this is a graduate student in the lab at University of Buffalo, in this lab, a researcher named Popescu. They were like, well, what's going on? And they were like, hey, there are these other NMDA receptors that are just kind of on all the time in some neurons. And we don't know why they should just be, NMDA receptors should be turned on with certain stimulation and then turn off again. And that's kind of part of their job. But there are some of them that are just turned on all the time. And so they're called chronically activated NMDA receptors. And they found that low doses of ketamine only attach to those chronically on NMDA receptors. And when they bind with those chronically on NMDA receptors, it changes the way the neurons act. So instead of being turned off and not wanting to talk to other neurons, suddenly the neurons, actually the other momentarily stimulated NMDA receptors and the serotonin receptors and other things in that neuron, want to do things and get activated. And so once they're activated, they start sending signals that neuron never used to send before, because there was an always buzzing noise that kept them turned off. And so when the low dose gets in there, it only fits the bill for those chronically on receptors, changes the way the neuron acts. The neuron then goes, I can be on. Okay, let's go. It starts making friends. And once a neuron starts making friends, it wants to keep making friends. And that is the rapid effect of a single dose of low dose ketamine, and also the continued beneficial effect of that single low dose of ketamine therapy, because it jump starts the neurons. It shuts down that noise that kept the neurons from being able to be friendly. Really, it shuts off the noise that was holding the neurons back. And they found that when they added too much or more ketamine, it dumped everything into the system, and they saw the sedative effect. They saw the dissociative effect. I don't know. I'm just very excited about this, because what it does is it reinforces the idea that, the quote unquote dose makes the poison, or the dose makes the therapy. And it also really gives us a new appreciation for what we thought was a very, relatively uniform population of NMDA receptors, to understand that there's now, why are these chronic ones on? What turned them on in the first place? Why are they different? Does ketamine have to be the thing? Or can we use other compounds that are just targeted directly at those particular NMDA receptors. Like this just gives us an opening into creating even better, faster acting, more efficient, effective medications for depression, especially for chronic depression that has been untreatable by many other therapeutics. So I'm super excited about this.
Making the untreatable manageable.
Yeah. I mean, for years I've been like, okay, so it does. Every other antidepressant that we know, it takes like a month for it to start working because it gets into the system. It has to get in there and start telling the neurons, the the neurons, the Padawans, to start expressing new receptors, right? Oh, you need to have more serotonin receptors to want to talk to the serotonin that's in between, right? You have to build it up. And so how do you do that? And it takes about a month at least for many of our antidepressants currently to do the receptor plasticity that's necessary to see change start to happen. But with ketamine, because of the way that it turns off something that is messing things up, it's like, stop that. Everything else can work the way it's supposed to right now. And then once everything is working, it does what it's supposed to. And so it just kickstarts something that should also already be working. So I'm just super excited. This is one of I haven't come across like a neuroscience finding like this where it's just so crystal clear in my head how it works. I'm just thrilled. I think this is gonna help so many people.
Nice.
And then I have 2 more really quick studies. If you like to work out, that's also good for depression, but it's also good for your muscles. And hey, it's good for your nervous system also. MIT engineers discovered that when you exercise, it doesn't just benefit the muscles, it actually benefits individual neurons. When muscles contract during exercise, they release myokines, which are signals, like muscle signals, muscle chemicals. And there's signals that when they're generated, they lead to nerve growth that is 4 times greater than when there is no myokine release. So anyway, exercise helps keep those nerves working. So this is another kind of it's another way of looking at m- muscle atrophy, where we think just the muscles are atrophying when you're not moving around. It's not just the muscles, it's the nerves as well. The nerves are not being beneficially influenced by the muscles if you're not moving them. Also, this is probably part of the entire feedback loop between the muscles, the nerves, the nervous system, and our entire physiokinetics and mental health that I'm going to add onto that as well. This was published in the journal Advanced Healthcare Materials. And so maybe this will help with, is one of the things that might help with leading to treating muscle injuries, where we seem to have areas of issue in that respect. And my final study, we're all tired, right? And you might be getting tired if you're up late with me, or you're up later on the East Coast, or up earlier, like Justin, who got up an hour earlier than he needed.
I thought we were still in the time difference, time zone and plate tectonics were all conspiring.
All conspiring. Well, this study came out of the IMT School of Advanced Studies, LUCA, and it was published in the Proceedings of the National Academy of Sciences. It's a very interesting concept of ego depletion. Have you heard of ego depletion?
No.
So this is where your willpower starts to diminish and diminish and diminish when you get more and more tired. So it could be because of physical activity, it could be because of mental, cognitive fatigue, but basically your willpower, or your ego, your psychological ability to make decisions, to interact with other people, it becomes diminished as a result
Is
of
that
fatigue.
willpower or is that just tired?
So tired is one thing, but the willpower. So there's tired, but you can be tired and still somehow come up with the willpower to keep going. We have all these stories, right? Hero stories. In the face of all odds, people awake for a week at a time.
So if
Also
you're
people who
tired
are,
and making bad decisions, it's because you had a lot of willpower. As
yes.
opposed to being like, I'm tired, I'm not even going to try this. That's no willpower, but no bad decisions made.
And actually, the willpower to say, I'm tired, I'm not going to try this, might be the healthier choice, because what this study showed in the literature, like we've been talking about willpower stuff, there's books on willpower for popular science, like willpower, take care of yourself, just practice, don't eat the M&Ms, eat an apple, blah, 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 blah. And it's very controversial. And a lot of the studies have been part of that whole psychological social sciences research that is not replicatable. And so the question is, okay, ego depletion, willpower stuff, is this even really a thing? And so this particular study I find interesting because they looked at EEG, so electroencephalograms of brain activity during sleep, and also in the awake individual. And they kept people working, doing cognitively depleting tasks during different periods of time, and then looked to see during wearing their EEG caps, measuring to see what happened to brain waves in different areas of the brain. They found that there were localized disruptions of brain waves, so that the active brain state that is normally there for the entire brain when you are completely focused and you're working at your top power started shifting, so that different parts of the brain actually were like locally asleep. So things like your frontal cortex, which is part of decision making, kind of effectively goes offline because you have cognitively depleted the energy. So the rest, your
I didn't
brain's
know that was
working,
possible. That
right?
explains so much. Oh my gosh.
You're awake, you're
Partial-
functioning, your brain is still going, but little parts of it are like, I'm out.
That explains so much.
It really does. And what they found is in their tests, they showed that people who were given greater cognitive depleting tasks and had these little localized sleep brain issues going on, they were really cranky and they made decisions that were not nice for other people. They were very self-serving and short-term benefit based. They were not more thoughtful, compassionate, and long-term consideration kind of things. So,
But most, if you do a survey, most billionaires will claim to only sleep like 3 or 4 hours a night. No joke.
my silence is agreeing with you. So this, so they went from the control group to the fatigue group, and looking at cooperativity to hostility. They found that peaceful cooperation dropped from 86% in no fatigue to 41% in fatigue. And they were looking at about 500 people in this study. So it's not like a 10-person one, this is not huge, but this is a very reasonable sample size of individuals. And they did find that these sleep waves, actual sleep waves in areas of the frontal cortex when individuals were fatigued, and those were more likely to have more hostile behaviors. And so the researchers say at the end of this here, overall, these findings have important implications for multiple situations in everyday life, including economic transactions, legal agreements, as they demonstrate that when the brain is tired, we may make choices that go even opposite to our own interest. As a matter of fact, this is what people do in most criminal acts. And with that, I think we've got all our stories in. Do you think
All
that
right.
do you think that does it?
Oh, we can keep talking, but yeah. You need to get to bed. I need to probably go back to bed, honestly. It is 7 a.m. here. The sun has not shown up.
It's 10 o'clock at night, and it is very dark here. It has been dark for hours. Where are you, everyone?
Where's
Is it light
the
or dark?
sun? Where did the sun go?
Where'd the sun go? One of my favorite science writer people I went to a science writer conference this last weekend in Raleigh, North Carolina. Andreas von Bubnow. I've known him for years, since we were like wee babes in the science writer community. And I said, "How are you? Where are you these days?" And he goes, "Somewhere cold and dark." I said, "Oh, where's that?" He goes, "Oh, there's lots of Germans." I'm like, "You're German?" He goes, "Yeah." I'm like, "So?" He goes, "I'm in Berlin." I love him. He's just wonderful. What a wonderful time to meet the people and see the
Or as
people.
we call it here in Denmark, in the warm, bright south of Berlin. Way down there, where they get the extra
Way down
daylight
there.
and the better weather. Yeah.
I love how you've taken on the Danish. Yeah.
Yeah.
Eric Knapp, 9:06 p.m. in Anchorage. Sunset was at 4:22. I love it.
Gosh. Yeah.
May it be a bright night and a sunny day wherever you are, whatever weather conditions there are. May you have bright lights in your life that lead you in the right direction and bring you joy. I want to say that is what you all do for me, and I am so glad that you've been here tonight. Justin, thank you for joining me to do this show. Last week, I had to cancel. I just couldn't hold it together, and I apologize to everyone out there because I was not in a place to bring my normal energy to the show. But thank you. And everyone out there, I want to give you the shout outs for sure. Chat room, you are my faves. Thanks for being there and chatting. Discorders, you know who you are. Thank you for being in the Discord. I know you are there as Patreon sponsors. And if anyone wants to be a Patreon sponsor, you too can join the Discord channel and chat. Fada, thank you so much for your help with show notes and with social media, and Gord and Arnlore for helping with our chat rooms to keep people on the up and up. Everyone also, actually, thank you for helping keep everyone who's in there. You're all this is our community. We do it together. Identity 4, thank you for recording the show. And Rachel, thank you for editing the show. And as always, I do have to thank our Patreon sponsors. So thank you to Eden Mundell, Alan Viola, Aaron Anathema, Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, George Corris, Pierre Velazarb, John Ratnaswamy, Chris Wozniak, Vegard Shefstad, Jonathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan Schubru, Sarah Forfar, Sarah Forfar, Don Mungus, PIG, Stephen Alberon, Daryl Myshak, Andrew Swanson, Fredis 104, Sky Luke Parwonovich, Kevin Reardon, Noodles, Jack Bryan, Carrington, David E. Youngblood, Sean Clarence Lab, John McKee, Greg Riley, Mark Hessen Flo, Steve Leesman, a.k.a. Z. McKen Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, I miss you, Remy Day, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, miss you too, John Atwood, Grudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Philip Shane, I haven't seen you in forever, Kurt Larson, where are you? Craig Landon, Sue Doster, Jason Old State Neighbor, Eric Knapp, you're in Alaska, Lon Makes, I think you're in the chat room, EO, Adam Mishkan, you're somewhere nearby, Kevin Parachan, Aaron Luthen, Bob Calder, Marjorie, it's been a long time, Paul Disney, I know, David Simile, Patrick Pecoraro, I've seen you on the internets, and Tony Steele, yes, same. The internets. Thank you. Thank you all for your support on Patreon. If you too want to support us on Patreon, please head over to TWIS.org and click on the Patreon link to become one of our ongoing supporters. On next week's show,
We will be back Wednesday, 8 p.m. Pacific time, and Thursday, 5 a.m. Central European time, broadcasting live from our Twitch and our YouTube and the Facebook channels and all the places where you get podcasts.
But you can listen to us a podcast too. If you want to just look for This Week in Science, wherever podcasts are found, we're there. And if you like the show, make sure that you get your friends to subscribe too, because who do you want to be? The only person talking to yourself about this stuff that we're talking about?
I keep finding it's new places too that I'd never even heard of before. For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. where you can also sign up for newsletters or listen to past shows there as well.
Yeah, and maybe there'll be a newsletter eventually, maybe for Christmas. We love your feedback. If there is a topic that you want us to cover or address as we head up to our 1000th episode. Oh my goodness, 7 episodes away. What? Beginning of the year. Get ready, everybody. Just let us know you can go to our social media accounts. Yes, we are on Blue Sky, Mastodon, and Facebook, or YouTube, or these Facebook and YouTube, whatever thing, and Twitch, whatever. Put TWIS in the subject line if you want to send us an email, so that your email does not get spam filtered into a neuron that has a chronically active NMDA receptor that's just like... Yeah.
We look forward to discussing science with you again next week. And if you've learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done.
This is the after show, and what we say will not represent anyone's views other than our own, and we say it in a way that is compassionate and kind, and is not looking to be a target of anyone at any time. Ta-da! I don't know where Justin went for this last little bit. He probably went to sleep. It's very possible. I have lowered my desk and my microphone for a couple of seconds here. We did it, Paul. Yeah, another great show. Woohoo! We made it through. Day by day, everyone, right? You know, kindness, compassion, humanity. We have different personal needs or wants in the moment. But when it comes down to it, we are all human. All human. More human than human.
Rob Zombie.
Yes.
Oh,
It was either it was either that or Madonna. You know how I work.
That I can also sing just as well. Actually, I can remember that one. This is fine.
We made it through the wilderness. No, we haven't. You know, we made it through. No, we haven't.
All right. So yes, Paul Disney, we did it. Another great show. I gotta go.
I do too. I have to go to bed. I think a
I
lot of
will.
people need more sleep. Okay, everybody? No 3 to 4 hours sleeping because Silicon Valley tells you that that's what you're supposed to do now. Okay? Be good to yourself.
No.
Rest. Take care
No.
of yourself. Let
Unless you want to
go.
become a sociopath. I think that's how they get there.
Right. Just yeah. What if people just slept more and took care of themselves and maybe that would like reduce a certain amount of environmentally induced sociopathy. Yeah.
Yeah.
This is a hypothesis. Maybe we should start with experimenting on it with Silicon Valley. Anyway.
We should do a 24-hour sleep show.
Instead of like the other one though.
Right. Yeah. It's like, these people do these 24-hour shows where they're trying to stay up all the whole time. We'll do one where we try to stay asleep for 24 hours.
And it'll
think
be
it'll be-
very chill. We'll see if we can work with one of the chill hop channels or something. And
We'll have a
it'll just
little
be
bit of
like the
chill music,
occasional snore.
some rain
Some
sound.
rain.
We'll put them all together. We'll have a dryer going in the background. We'll put all the sort of sleepy
Oh.
sounds on at once.
Oh no, this is the new channel, Justin, and this is gonna be really big.
Oh, it is.
24-hour Higa.
I'm telling you, those channels exist already, and they are
But
cranking in multi-millions of
I
viewers.
really think this is like maybe we can put like sleep facts up and stuff like that. But most or like social connection and cozy time, science-based facts. It'll be like, yeah, it's our 24-hour Higa.
This
TWIS
is
Higa.
like the only word things. Yeah.
It'll be like a playlist,
You're
or we can do it.
sleeping so good.
Yeah. Oh my God, I
Yeah.
love this. I could make this. 24 hours of TWIS Higa. Higa. Am I getting close or am I totally off?
It's fine. Yeah, I'm not sure that the Danes know how to
Pronounce it themselves.
pronounce their own language exactly the same because there's regional dialects in a country with 6
Everywhere.
million people.
It's amazing.
So I don't know that there is a right. And then the Copenhagen dialect,
Copenhagen.
take all the words and turn it
Yes.
into one monotone, quick sound.
So the whole language is a soundbite,
Yeah,
but like a temporal soundbite.
it kind of is. The Copenhageners get rid of all of the spaces and they shrink the words to just enough of an inflection of the sound that you know what word it was supposed to be, or you think you do.
Well, yeah, you're good luck with that. Keep going. Fighting the good Danish language fight, whatever. Everyone out there, we're gonna struggle
Everybody
together.
sleep.
Everyone sleep. Yeah. Say goodnight, Justin.
Can't.
Oh, say good morning, Justin. I thought you were
Good
gonna go
morning,
to sleep.
Justin. Say good night, Kiki.
Good night, Kiki. Good night, everyone. Thank you for once again joining us for an episode of This Week in Science. And we do hope that you will join us again next week for another one. You'll be back, right, Justin?
Yeah, it should be. Why wouldn't I? What do you know?
I don't like that answer.
What? Yo, can I just say right here at the very end?
Just say
Hang on. I pulled up this story. And it said, death god of chaos asteroid to possibly
I thought
hit
about
Earth
that one.
13th of November. And I was
thought
like,
about that one. I was like,
well, that's today.
Nah.
But it's 13th of November, 2029, which still, but
Today.
But it was
4
it
years,
said the
5
13th
years.
of November, so I was like, oh, well, I'm not going to bother taking out the trash. No point in doing laundry now. I had all these cleaning plans.
No, you still have 5 years.
I still have 4 years to do it, so I can still put it off.
You have 4 years, 5 years to do your
5 years.
laundry. Fantastic. That's a
It's going to be 4 years till I run out of underwear.
Oh, God.
So much underwear.
Because do you have it on
Whole
mail
closet.
order? I don't know. Okay. Good night, everyone. I really do
Alright.
hope that you stay well, stay safe, stay curious,
Stay lucky.
stay lucky. Yes. Each of us individually can be lucky and curious and all these things.
I'm just going to put at the end of all of them from now on.
Except for
Need more.
stay connected and support each other.